My name is Gustavo S Jeuken. I'm a PhD student at the KTH Royal Institute of Technology in Stockholm, Sweden, and I'll be presenting my poster entitled Survival Analysis of Pathway Activity as a Prognostic Determinant in Breast Cancer. This figure gives a brief overview of our analysis, and I'm going to go carefully through it. We start with our sample dataset. We downloaded this dataset from the Metabolic Consortium, and it contains both gene expression data and clinical data of over 2,000 freeze-dried breast cancer samples. The second data source we use is the Reactant pathway database. That will serve as the group of gene grouping that we use later on our factor analysis. We use the pathway database to subset the gene expression data. That is, for each pathway, we will only use the gene expression information for that specific pathway. With that gene expression information, we run our factor analysis, more specifically a confirmatory factor analysis. So, having only the gene expression data for each pathway, we perform principal component analysis and use the first principal component, that is the direction where those genes are most varying on the gene space, as the most likely to be representative of the activation of that pathway. So then we project the samples into the first principal component to get what we think is the best proxies for the individual pathway activation of that sample. Then, having the individualized pathway results for each pathway and the clinical data, we combine both in our survival analysis step. Here, we perform a Cox regression by regressing in the individualized pathway results and the clinical data for each pathway separately. That is, for each pathway, we input the individualized pathway results of every sample against the survival data we have from the clinical information for the same samples and get our results. Then, from those results, we extract the pathway prognostic significance. And this can take mainly two forms. The first is a p-value associated with the regression coefficient for each of the pathways. And the second way we can get a pathway prognostic significance is by doing a cross-validation. That is, setting apart some of the data, running our regression on part of the data, and then seeing how well that regression predicts the survival of the rest of the data. We can do this several times for different parts of the data and then average the results to see how well it predicts the survival of the data set. A bit more specific on the factor analysis part, we try to leverage the fact that gene expression data is often correlated. And as an example, you can see here a correlation plot of the genes in the regulation of TP53 activity. You can see there are some cluster of genes that are positively correlated with each other, but also genes between those clusters are negative correlated. That is, if there's a high expression of genes in this cluster, there's a negative expression of genes in the other cluster. And by doing PCA and taking the first principal component, we hopefully leverage all of this information. You can see another good example here. We look at the survival plot for the gene CA9 membrane protein. By our Cox analysis, this gene has been found to be significantly associated with survival on the samples. On the right, we have two pathways in which this gene is present. One is regulation of gene expression by hypoxia-inducible factor, and the other is reversible hydration of carbon dioxide. On the first pathway, most of the genes are varying in the same pattern as the CA9 membrane protein. Therefore, you can see an increase in relevance associated with survival. However, in the reversible hydration of carbon dioxide, the genes don't follow the same pattern as the original gene, the CA9 membrane protein, so we see a decrease in the association with survival. Over here you can see the top 10 pathways that have been found to be most predictive of survival. They are ranked by concordance index that was obtained on the cross-validation. Also displayed are the Q values from the regression coefficients, and those have been corrected for multiple hypotheses. And finally, we have implemented an easy way to visualize all the results using a sunburst plot. The advantage of using a sunburst plot is that it displays the pathway hierarchy in a very easy manner. This is interactive, and you can navigate through the different pathways by going down or up the hierarchy. You can see all the associated statistics on the pop-up bubble, and you can also see, for visualization purposes, the Kaplan-Meier plots that are mean separated. Thank you for listening to my presentation. Goodbye.